Hello, everyone. Thank you for coming. So in this half an hour, I'm going to try to talk to you about the protein X-ray crystallography facility and a bit of AlphaFold. The facility is in room 1S205. I'm the manager there. I'm supervised by David. We share the room with the crystallization facility, which is run by Fabrice and supervised by Jan. So what is protein crystallography? Basically, you need to crystallize your protein. Then you're going to hit it with an X-ray beam. You're going to get a diffraction pattern. From that, you can use Fourier transform to reconstruct your real space shape. In this case, it's going to be your electron density. And then you can build your model. Since I'm going to talk a little bit about Fabrice, you couldn't do it today, so I'm going to do I've got 3 or 4 slides about the crystallization facilities. What is required for protein is requiring very pure sample, and ideally 1 milligrams of it. And to crystallize it, you're going to mix your pure sample with some reservoir, and then by vapor diffusion, the water is going to go back into the more concentrated reservoir. The drop is going to shrink, the protein concentration is going to increase, and if you're lucky, you will get some crystals. Crystallization occurs in the nuclearization zone, where first it nucleizes, and then moves back to the metastable zone, where the crystal can grow. This is what, in an experiment conducted by Mark, Fabrice finds this nice drop where you can actually show all the faces. And this is where the crystal is starting to grow, become big rather than staying small. And that's what the type of crystal you're looking for. So in the crystallization facility, Fabrice uses the plates designed the LMB, which is called the MRC plates. They got 96 different conditions, and he has 23 different plates. So together, you got over 2,200 conditions to get lucky. And it's a game of numbers. The more you try, the more chance you have to crystallize a protein. It's all done by automatic robots, like the Mosquito. We set up the plates for you. Or you can even use this days you will go dispenser, Heco dispenser to do nano drops in case you're very limited in the samples amount. But it's quite fast. It takes about, I think I said somewhere, yes, about 5 minutes per plate. So if you need to set up all 23 plates, you got 2 of these robots working at the same time. It takes you probably an hour. Once you have a crystal, you need to harvest them. We harvest them into this kind of loops here, which are attached to this pin and this magnetic base, which is going to go on the goniometer. That's what the nano loop looks like. And also, there's different type of lithographic type of loops. You use this magnetic wand to attach at the bottom of the base to help you in fishing. That's what it looks like. This is what it looks like when you're trying to fish a crystal under the microscope. Once you fish your crystal, you quickly flash freeze it into one of these packs, which will be submerged in liquid nitrogen. Like this. With the magic wand, you can just push it out of the wand. Then you close the pack, and you can store them infinite time. Or you can ship them to the synchrotron, as we'll see in a bit. So if you want to if you want to collect in the house, we have an X-ray generator. This is the rotating copper anode, which will generate 2 sources of electron, one this way, one the other way, with 2 independent detectors. So you can have 2 people having experiments at the same time. Oh, we can ship to the synchrotron, which is a large ring where the electrons are moved at the speed of light, and when they change direction, they release X-ray, a large amount of X-rays. They go all sorts of MX beamlines, macromolecular X-ray beamlines, to do different type of experiments with different crystals. Oh, sorry. And if you need to send your sample to the synchrotron, we can arrange it together. There are 2 ways to do it. One way, you can physically go to the synchrotron, which is a nice experience. You can see how the beamlines are inside, how everything works. Or you can just collect remotely from the X-ray facility. We have a setup downstairs with a table and 2 screens. Again, if you need to do anything of this, we can arrange it together. I will help you. Just a little briefly about crystals. So crystals, the arrangement of the molecules, which is all in the same direction inside the crystals, is defined by a lattice. And these lattices that can produce crystals, there is only 14 type of lattices. They're called the Bravais lattices. And that's all I will say about this. So why we need crystals? Because imagine if you have like a single molecules, and you hit with X-rays, you will get the scattering, you will get the scatter from a single molecule, but you won't be able to see this because the interaction of the X-ray with the matter is very little. So we don't have sensitive enough detectors to see the image from a single molecules. So we use crystals which are able to amplify this the signal like a billions of times, whatever the number of molecules are in the crystals, due to the lattice. However, there's a problem that the diffraction will only be in the direction of the lattice points. So it's only where you see the point it will be amplified the signal, and in between the points the signal actually will be destroyed. There will be no interference. So what you will end up with an image of your molecules, but only in the direction of the lattice of the crystals. In reality, when you shoot a crystal with an X-ray, that's what it really looks like. Each of these points is called a structure factors. Every structure factors has a amplitude F, a phase alpha, and 3 indices, which are called the Miller indices. Now, to reconstruct, okay, this should not appear right now, but it's okay. To reconstruct your, actually, no, it should have. To reconstruct your density, any real space three-dimensional object can be approximated by a Fourier series. In this case, the Fourier series is the summation of all the structure factors that you collect in an experiment. So you have here, you need all your amplitudes, your phases, and your Miller indices. Once you have all this F, alpha, and Miller indices for all the spots, you can plug it into the equation. You're going to have your real space three-dimensional object, in this case, the electron density. Now, the more spots you can collect away from the center, the higher your resolution is going to be. This is an example. If you got this number of spots from a diffraction, you're going to get some maps look like this. If you get this type of spots, this amount of spots, your maps are going to look like this. It will be much easier for you then to build your model out of it. Very quickly, how do we find the amplitude, the Miller indices? So let's pretend we have collected the data set, rotating your crystal 360 degrees. We use a spot finding where you can find where all the dark spots are, just digitally. Then with the information you got from the image header, you got the vector distance, beam center, and the wavelength of your X-rays. You can reconstruct the reciprocal lattice in 3 dimensions. So imagine these are all the spots that you've been collecting on a single image, put it all together and reconstructed or aligned. You can see in a minute, you should be able to see. If you line in the right way, you actually be able to see the lattice. There is the lattice, just to show that it exists. So once you have the lattice, you can start to put your Miller indices, 1, 1, 1, 2, 2, 2, 3, 3, 3, so on. And that's called the indexing. So once you've done the indexing, now you can go back, you can measure the intensity of your spots, because you can predict where your spots is going to be. And then if you take the square root of your intensity, you're going to have the amplitude. So now you got the amplitude, you got the Miller indexes, all you're missing is the phases. To get the phases, there are different methods. Molecular replacement today is one of the most popular, thanks to molecular replacement models coming from AlphaFold. There's no other way to find the ab initio phases, but that's all I'm going to say about this. Once you have all your components for your equation, you plug in, you get your density. Hopefully, you had enough parameters to get a high resolution. So you can start to construct, if it looks like this, is quite easy. You already see like a tryptophan there. So you can start to build your model. And usually this is done in software called CUT, which is developed by Paul at the LMB. Then you have to do some refinement, which is done using usually RefMax, also developed by Gary by the LMB, or Phoenix or Buster. And then you have to do the validation of your model, checking that all your chemical angles and bond lengths are sensible. All the software you need to do what we just showed you is all available on the LMB server. If you want to know a bit more, I can recommend you some books. But also there's some nice videos of a lecture from the people that actually wrote the software that you'll be using. So I recommend to look at those videos. And me and Fabrice are always happy to talk to you. And also, if you're interested, there are some workshops. There's a two-day data collection at Diamond. And also, there's a 7-day, like the full Monty from A to Z. If you're interested, just let me know. I will contact Diamond and find out if there is a spot available. Just some examples. So before AlphaFold arrived, this is some of the work we used to do at the facility. So we structure like in low resolution maps from a cryo-EM. These were low, so people, they couldn't build here because the resolution was too bad. So they used to just crystallize the small part of the protein and then plug it into the density. This is Ralph, John, and Zigu. These days, this is pretty much done from AlphaFold, as we will see a bit later. So what people are doing these days is things that you cannot do with AlphaFold, like RNA molecules, phosphorylated, phosphorylation, post-translational modification. Yeah, there's small ligands, like a drug discovery, or unnatural amino acids, which AlphaFold cannot deal with. And from that, I'm gonna tell you a bit about AlphaFold. These slides were kindly donated by Sami, who's gonna give a talk in February, an in-depth course. I'll just give you a little introduction today. But also there is a very nice talk from Sami he gave back in March about these lectures. I strongly recommend you to go and look at it. So, AlphaFold. What's AlphaFold? AlphaFold is one of this new AI. Probably you know already, but I'm going to tell you. It's an AI tool which is based on a neural network which has been trained on the protein data bank, which means that all the structures have been solved experimentally, either with X-ray, cryo-EM, or NMR. And it also uses the co-evolution system, which from the multi-sequence alignments, basically takes a multi-sequence alignment from homologs and looks for residues that have been evolving at the same time during time. So he thinks because they've been evolving together, they must be close to each other in the structure. That's very roughly how it works. And of course, it's not the only one. There's also Rosetta Fold, which work in a similar way. And also, there are some protein language models like ESM Fold, the Omega Fold, where the multi-sequence line is already embedded in the algorithm. For that reason, they work much faster. However, they are not as accurate in the result as AlphaFold or Rosetta. And between AlphaFold and Rosetta, basically, if you have to pick one out of this, probably you will pick AlphaFold. Overall, is the one that performs the best. The input is just your sequence. You don't need to purify protein. You need to crystallize nothing. Just an email from somebody. And the outputs is going to give you the top 5 hits that he thinks are the best results. Also, going to give you some metrics for the multiple alignments you used. It also give you some confidence predictions. There's a per residues confidence, which is used for the secondary structure, and a pairwise confidence used for the interaction between domains or between different proteins. So the multiple sequence alignment, as you can see in this case, is being used in like about 3,000 different these sequences. Rule of thumb says you need at least 30 sequences, which seems very easy. But sometimes if you have a super divergent protein, that could be a problem. But 99% of the time, it should be okay. As you can see, near the end, near the beginning and the end, they're usually at different lengths. They're shorter or longer. So it's never great. But usually in the central part of the protein, the alignment is quite robust. So this is just going to show you that it's been using enough sequences. So for you to see, oh, okay, it's been using 3,000 sequences, that's plenty. So I don't need to improve anything in that area. Like we said before, this is the per residue confidence. If you are below 50, means usually it's a disordered area. If you are above 50, there is a secondary structure there, like a helix or a beta sheet, like you can see in this case, helix, unstructured areas. And then there is the pairwise confidence. This is going to show you. This is the error between a residue and another residues within the problem. Basically, you put all the residues here from 1 to 400, here 1 to 400, then makes a correlation between each residue against each other residues. Of course, in the diagram, it's going to have the best score because it's each residue against itself. So it's in the same position, they're overlapping. But for example, let's see what usually if you have a well-folded domain, like in this case, this green domain here, you're going to have a cluster or blue spots here. Say, look, I'm confident this domain is well folded. Same for the other domain here. But you see, if the two domains are sitting next to each other, then you're going to get also these two clusters, which is between the interaction between this domain and the other domain. This residue interacting with this one, this residue interacting with that, just because they're located very close to each other. Now, they're not the most easy things to interpret, especially when you start to have bigger structures. But there are some tools, like there's a plugin in Camera X, which is going to help you with it. It's all color coded. It makes it a bit easier. There's also a web server where you can upload your result, and it helps to look at to interpret your validation reports. AlphaFold, you probably know, there have been a few versions so far. So the latest one is AlphaFold 3. But we are still using AlphaFold 2, and we'll see soon why. AlphaFold 3 is able to handle different types of molecules, like protein, DNA, RNA, ligands, and ions. It's faster, much faster than AlphaFold 2. You can accept 5,000 amino acids. AlphaFold 2 usually takes 2,000. There's only the online server, though. We cannot run it on our cluster, or at least not at the moment. It's not straightforward to set it up. So at the moment, it's only the one available online. You are only limited to 30 jobs per day. But however, apparently produce more hallucinations. So it can get some false positives. AlphaFold 2 only handles proteins, but it's very customizable. So you can try to, you will see what Sami's talk, you can kind of trick it to find different conformation for your proteins and other things. It can be run on the clusters. Apparently, it's more accurate for multiples. That's what was reported by the LMB users. So AlphaFold 2, there are two ways to run it. One is through the Colab notebooks from Google, which is online. Again, you just apply your sequence here and you run it. It probably takes an hour. Oh, you can run it on our cluster, which has been set up by the scientific computer together with Sami. AlphaFold 3, as we said, there is only the web server. You can use not just protein, but you can also use different, you can add the ligand, you can add ions. I don't know how well performs, I must admit. I had very mixed reviews. But the nice thing is you can also handle bigger structures. So if you do a bigger structure, probably you can use AlphaFold 3 because, as I said, you can go up to 5,000 residues. However, when the structure gets very big, it probably is the best strategy to do fragmentation. Like in this case, for this dynein protein, if you just put the whole thing into AlphaFold, you come up with this fold, which is not right. But it's possible to fragment and get it bit by bit, and then just rebuild your full complex later. There's another example. If you put the full length, it doesn't look right. But if you split into 4 pieces, you get all 4 correct pieces, and then you can just, because you go overlaps, you can just then reconnect it. So most popular ways why people are using AlphaFold so much, as we said, I showed you before, with crystallography, we used to crystallize like small domains which were not well resolved in cryo-EM. Now that can be done with AlphaFold very quickly. You can see this is a very nice fit to this, and so on. And in other ways, you want to if you need to do some biochemistry with your protein, it's insoluble. You want to try to understand where you should cut it. So, for example, this is an example from Sami. He used to cut this gene here. His protein was insoluble. After he ran AlphaFold, he realized that he was destroying this polylicos bundle. So he moved a bit further here. He started to cut the protein here, and in that way, was able to create a nice soluble protein for his biochemical experiments. Variation on the team. So then people have been taking the AlphaFold algorithm and try to do some clever things with it. A guy called from the Kosinski lab, they designed this AlphaFold pull down, which is able to use AlphaFold in a way that you do pull down assays. For example, you give it a target, and then you give a population of protein. AlphaFold will check which one is interacting with which. Somebody did something similar for the LMB. They set up what's called alpha screen. It's just like alpha pull down. It's found on the cluster. You can just choose your target and then give a list of UniProt sequences. It will do the pull down experiment for you. However, the results are not easy to interpret because you can imagine when you're going to have like thousands or hundreds of thousands of this to decide which one is real, which one is not. It can be tricky. There are some ranking numbers, like the IPTM, the list. I won't go through this. Some we will explain to you in February, but it's still tricky. Another way to use AlphaFold has been something called Alpha Link 2, where as well as the alpha code, as well as the sequence, you can also input your cross-linking mass spectrometry data. So where the cross-linking mass spectrometry is going to tell you which residues switch to another one, and that's going to help AlphaFold to predict the structure. For example, this is the crystal structure. If you only gave the sequence to AlphaFold, apparently failed, but when you supply the cross-linking data for cross-linking, he succeeded. Yeah, okay. And also there is this bind graph. This was developed by a guy called Pasisa, and he uses the AlphaFold algorithm at reverse. So you give him a target. You say, I want to find the product that's going to bind here. He's going to go through some iteration. It's going to find the product that's going to bind to the side that you wanted. And also, it's going to do, after he found, let's say, he found 50 different binders, he's going to run a solubility test on these 50 binders. So he will tell you which is the most likely to be soluble, because once you create it, probably you want to express it. So in the past, the problem was that you were getting all these hits, but then when you try to express it, they were all like insoluble. They were all like synthetic domain, synthetic folds. This one apparently works a bit better, but the success rate is still an open jury because it's a very modern algorithm. Also, it's worth to mention that there are structural homology algorithms at the LMB, like Progress, done by Greener and Jamali here at the LMB, similar to Fold 6 and DALI. Basically, you can supply a fold, and then we'll find you all the protein with the similar fold. This can be useful when you're working, for example, in tomography. It should go very, very, very low resolution. Okay, limitation advanced use cases. Isoform specificity is a problem. Let's say you got two proteins which are isoform of each other. One isoform is binding to your kinase. The other one is not supposed to bind. If you put it through AlphaFold will show you that it's going to bind, just because there is some biases towards the other isoform, which is also in the PDB where it was trained. Also, recombination state is a problem. Let's say, in this case, you knew that your protein is a dimer, but if you didn't know and you feed AlphaFold 3 copies of your protein, it was still going to create a triple helix, which looks, it looks like it could exist, but we know from biochemical experiments that it is a dimer protein. Other problems are like, alternate conformation are difficult to get. Usually, AlphaFold will give you one conformation, either the closed one or the open one. But we're using AlphaFold 2. AlphaFold 2 is possible to customize it. You can change the number of seeds. You can change the depth for your multi-sequence alignment. You can add the dropout. And people claim that sometimes you have been able to pull out the second conformation for the protein. And also, there are some specialized algorithms like AF cluster to find the different conformation. I never use them, so I can comment how well they work. Post-relation modification is a problem. Of course, if you have like a phosphorylation, you cannot give the phosphorylation to AlphaFold, so you will just leave a gap there, but you still might be able to position your peptide in the right position, but you will have not the phosphorylation. And mutation is also known to be a problem in AlphaFold. If you mutate, doesn't matter what you mutate, you're still going to get back exactly the same structure if you know that that mutation is affecting the conformation of your protein. Apparently, Rosetta Fold is better in that case, but I have not tried myself. Ligands nucleized used to be a problem, as we said, because AlphaFold 2 cannot accept protein. But these days, we can have AlphaFold 3. Also, there is Rosetta Fold Olatum, which can accept ligand and nucleic acids. Again, how well it works, you'll have to try yourself. Otherwise, in the past, we used to have AlphaFill and Alpha2 bind, which you just give the empty protein from the apoprotein from AlphaFold, and then you tell it that you want this ligand there, and you try to fill it according to what you can see that is already in the PDB. Antibody is also a problem because we said that AlphaFold works on this coevolution of the residues, but antibody are designed to evolve very fast, and that confuses AlphaFold. But there are some specialized algorithms for that. You can try AlphaFold in that case. And as we said, probably very low homology is a problem because of the multi-sequence alignments. In that case, you might want to try the protein language model. algorithms. Although these have the multi-sequence line is embedded in this one, so they will also struggle if you go very pure homology. Of course, there is a database of AlphaFold structures, so you might not need to have to run your own AlphaFold job. You can just first of all go to the AlphaFold protein structure database and see if somebody already done it for you. The only problem, you will only find monomers. If you know that your protein is going to be a dimer, you will have to run an AlphaFold multimer job yourself. On the resources, there is an AlphaFold list. Sam is in charge of this, but he's always posting some new updates. So you might want to sign up for this list if you want to stay up to date. And I mean, this is some nice papers. This is very nice, how to interpret AlphaFold structure, it helps to understand how to look at those graphics, like the confidence reports. Collab from Google, we already mentioned that. Thank you for your attention. If you have some questions, I'll be very happy to answer.
Okay. Hi, everyone. I'm Jane. I am a support scientist in the NMR facility. And I'm going to be talking to you today about what we can do with NMR to help you understand your structure. So we heard about interactions from Trevor earlier. So now I'm going to be focusing on what we can do with NMR and looking at structure. So just to summarize the kind of things I'm going to be talking about. So I'm going to talk to you about how we would traditionally calculate an NMR structure of a protein, talking to you about protein dynamics. I'm going to follow on from Dom's talk and mention a tiny little bit about AlphaFold, but then I'm going to talk about some of the main uses that we find people are using NMR for here at the LMB, and that's studying phosphorylation and also looking at intrinsically disordered proteins. And I'm going to take one slide to talk about AI because that's super fashionable. So, depending on, I don't know what your undergraduate background is, but if you've come from a chemistry background, you probably, when you think about structure by NMR, you're thinking about calculating or confirming the structures of small molecules. And that's something that happens here in the LMB, for example, from Southern Lab, looking at derivations of adenosine molecules. But then that's not the only small molecule that we look at. Trevor alluded to looking at glycosylation sugars yesterday, carbohydrates. You might not know that NMR spectroscopy is probably one of the most accurate techniques of identifying non-Watson-Crick, non-bDNA secondary structure elements in DNA. For example, I motifs and G quadruplexes. This is worked in the SAIL lab, because these base pair hydrogen bonds have unique chemical shift perturbation, chemical shifts in the proton dimension, which mean you can identify these secondary structure elements. But the bread and butter of the work that we do in the NMR facility is looking at proteins, and that's proteins in the solution state. And this is really important. The size of the protein is quite important to the kind of information that we can give you. So as it's probably quite well known, traditional solution state NMR is really best suited to smaller systems, so up to say 40 kilodaltons. But as a general rule of thumb, the bigger the protein, then the more complicated the data collection and the more complicated the sample preparation is in terms of the way that you produce a sample with isotopic enrichment. And then also, as you get above 40 kilodaltons, then the completeness of the data set, the data coverage we can give you is also reduced. But that's not to say we can't tell you about bigger systems. I mean, we have had looking at a system that's half a megadalton in the magnet, but that information is based on very specific questions that we're asking, and then you need specific labeling. So all this is true for a globular or a folded protein, but we also can look at proteins that are intrinsically disordered or sequences intrinsically disordered regions of proteins by NMR, and then those previous rules don't really apply. We can comfortably look at IDPs of 40 kilodaltons or more, and we don't need to have these super complicated labeling techniques. And we also have equipment in the NMR facility that's specifically set up for looking at these intrinsically disordered systems. And perhaps this structure of an IDP is a little bit misleading to start with, because, you know, some IDPs have small regions of secondary structure element, but they're not, these loops are disordered. And so if a more realistic interpretation of the structure of an IDP is this NMR ensemble, where you can see that the termini are flexible and they adopt these different orientations. So I mentioned it slightly on the last slide, so I think now is a good opportunity to just refresh what our sample requirements are for your NMR sample. So we ideally need your sample to have been expressed in E. coli recombinantly in minimal media. So that's minimal media you can order from the kitchens here at the LMB, and then you need to supplement that either with 15N ammonium chloride as the sole nitrogen source, and if you want it double labeled, also 13C glucose or deuterated 13C glucose from as your sole carbon source. We have stocks of isotopes in our facility, so if you start an NMR project, obviously come and talk to us about what it is you think we can do for you by NMR. We'll have a conversation about sample experiments, sample design, and we can give you the isotopes so you can get going straight away. If you want it deuterated, you can even grow your sample in D2O, which is quite fun. So again, sample conditions. For structural work, we're probably asking a bit more than 20 micromolar, but starting, say, 50, 100 micromolar up to 500 micromolar of your sample in an aqueous buffer, 500 microliters if you can. If you can't, we can do smaller tubes if it's absolutely necessary. We're looking at a mid-range pH for your buffer, about 1 pH unit away from your PI, ideally. And then before it goes in the spectrometer, we add 5% by volume of deuterated solvent, usually D2O, and this is just to help experiments work. We've got lots of protocols as well available, so don't think you have to try this without any help. We can guide you in expressing your proteins. So, traditional NMR structural calculation. Well, you do this in silico. It's called simulated annealing. You take your primary sequence and then all of this known restraint information about bond length, bond angles, etc. And then you add in our experimentally derived restraints. There can be thousands of them. And it's worth noting here that all of these restraints can be derived by NMR experiments. NMR isn't like a one experiment, one answer technique. There are hundreds of different NMR experiments we can collect that can give you lots of different intricate information about your system. And so we bring all of these together for our solution structure determination. And then what we want to do in silico is reconcile all of these restraints with a folded structure to make sure that they're in the final structure, you don't have any violations. You don't have any, the structure doesn't counteract any of these restraints that you've given the system. So one thing that is quite interesting as a side note, I don't know if anybody's ever downloaded an NMR structure from the PDB, but you'll notice that it isn't one single structure like you can get with crystallography or cryo-EM. You get an ensemble of between 20 and 100 models. And that's to do with the way the structure's been calculated. So as I said, it's called simulated annealing. What you do is you, in silico, you add lots of energy to the system with our restraints, and then you slowly remove the energy from the system. The protein folds up, and then what you want to do is end up in a global conformational minima, but obviously there are more than one minima in that system. So how do you know you're in the right minima? You do this hundreds of times, and then you should have an agreement that you're always going into the same minima, then you end up with the same, you have confidence that your fold is the right global minima. I think one thing that is quite interesting when you look at your structure is that obviously the core of the protein, you have a very close agreement, very small RMSD differences in the core, in the secondary structure elements, but you can get some heterogeneity in loop regions in the termini, and that can be simply because there's a lack of restraint there. There aren't that many restraints that we have for those termini, so you can start to see that perhaps these regions are sampling more than one position in three-dimensional space. I've said all of that about how proteins are calculated traditionally by NMR spectroscopy, but you know, structures deposited to the PDB calculated by NMR actually peaked quite some time ago in 2007. And you know, I'm going to say something that's probably quite counterintuitive from an NMR spectroscopist and say that I'm not going to stand here and say you should calculate your structure by NMR. There are lots of other faster, easier ways of getting your structure, but NMR is still a key tool for structural biology in the LMB. And why is that? I mean, it's a changing landscape, and I think it's because what NMR can do is tell you really interesting specific things about the dynamic properties of your protein. Proteins aren't static entities. We saw that with the ensemble of the NMR structure on the previous slide. They can experience motion on a very wide range of timescales. And a lot of those motions are going to be functionally important for that protein's role. And as I said before, there are lots and lots of different NMR experiments that you can run, and there are quite a lot of different NMR experiments that can probe specific timescales of motion, so you can have a window into quantifying the motion that's happening in your sample by NMR. So what's a quick experiment we can do to tell how dynamic my protein is? So here's a protein. It's got a disordered N-terminus and a structured C-terminus. And this is an experiment called the heteronuclear NME. So we have information for each non-proline residue because this is a HSQC-based experiment. And we can see we get a value in the heteronuclear NME. If our residue has a value of between 0.8 and 0.9, then we know that that residue is in an area of secondary structure element. It's in a folded system. But then the lower the value, or even negative you get, then that indicates that the protein is, the residue is in the disordered part of the protein. And this heteronuclear NME tells you about motion on one timescale only, and it's on the picosecond timescale. What I think is really cool about this protein is that even though you have an unstructured N-terminus, you do have these little islands of increased rigidity in that disordered part. And we know that these residues are involved in ligand binding. So even though you think you've got disordered N-terminus, there are parts of that disorder that are experiencing segmental motion that are important for downstream binding. So the two other main NMR experiments that you might have heard of for looking at NMR dynamics are the T1, or the longitudinal relaxation rate, and the T2 transverse relaxation. I'm really not going to say anything more about this at all, apart from to say that unlike the heteronuclear NME, the T1 and T2 relaxation rates are affected by motions that happen on more than one timescale. But what's interesting is that the T1 data set is T1 data can be influenced by the overall shape of your molecule, so whether it's anisotropic or isotropic, and your T2 is heavily influenced by the overall size of your system. some cool things you can do with your T1, T2 data. You can do this T1, T2 against T1 plot, and I think it's really useful. And this is an example for ubiquitin. So to read the plot, you have these red lines. So this is an order parameter S squared. And so the higher the order parameter, the more rigid the residue is. So you get 1 dot per residue. And then these blue lines are tau c, that's the tumbling time in nanoseconds. And so you can see the majority of the residues occupy the same part of the plot. So they have a correlation time of about 5 nanoseconds, and they're about 0.8 S squared, so that's rigid. But you can see you've got a number of outliers. And we know these are from the flexible termini of ubiquitin, and they're from flexible loops. And what's really interesting, if you look at the same kind of data, but for an IDP, you can see that now, so this is alpha-synuclein data, you don't have that dense packing of your T1, T2 data. It's all spread out. Your data has got a much lower order parameter, and you don't have one single correlation time. And that's because it's flopping around and also tumbling at the same time. So I think that's a really interesting way of having a very visual analysis of the dynamic properties of your protein. What's the practical application of that? Well, here's some T2 data that we have from a protein from Crystals, who was in the Modis lab, and he couldn't crystallize his protein. He didn't know why, so the protein was assigned and a T2 data set was collected, and we could see that there was this patch of increased flexibility, so a very flexible loop in the system. So Chris then truncated out the loop, and then he was able to crystallize the system, and that loop would have been in this part here. So very quick application of what T2 can tell you. So Dom spoke about AlphaFold, and I think this is a really, it's such a useful thing to do. Do an AlphaFold structure instead of doing an NMR structure. Why not? It's very accurate. One thing when it comes to proteins with multiple domains, you can see in this 3-domain protein that's part of a ribosome maturation assembly, you can see that AlphaFold says with some degree of confidence that the relative orientations of the 3 domains are, almost this. They're not confidently the same, but there is some degree of relative orientation between them. But when we look at our NMR data, actually paints a different story. So we can look at we can predict what the T1 data would be based on the shape of the molecule. And that's what we're seeing in these black and grey lines. If we predict it's rigid across the backbone, then we would have T1 data along here. And if there was quasi-independence, then the T1 data would be up here. And what you can see is that the domains 2 and 3 are tumbling together as one entity. So that means that their relative positions are going to be fixed, they're the same. Whereas the first domain has some pseudo-independence, and it has a different overall T1 value. And you wouldn't have known that from the AlphaFold assessment of the relative positions of the domains. And what's really interesting is that there are disease mutations associated with this protein, and then a lot of the disease mutations happen within this interface. And then a follow-up study by cryo-EM showed that the movement of this domain was important for helping knock out an inhibitory subunit. So if it didn't have this motion between them, it wouldn't be able to knock out the subunit and allow downstream maturation of the ribosome. So I'm going to talk about phosphorylation effects. And there's a really cool story looking at the phosphorylation of ubiquitin. Now, I guess you know about ubiquitin. Ubiquitin can be used as a post-translational modification to a lot of target proteins, and you can get ubiquitin chains, and you can get lots of different chain types because ubiquitin can be linked together at its C-terminus to all of these different lysines, or also to its N-terminal methionine. Ubiquitin can also be phosphorylated, and it can get phosphorylated at serine 65 down here. And in this project with Toby, who was a PhD student in the Commander Lab, he wanted to understand what the structural consequences of phosphorylation were. So we can do in situ phosphorylation, so you put your kinase in to the NMR tube and then watch the phosphorylation happen in real time. What was really interesting when we phosphorylated ubiquitin was we ended up with apparently 2 different species. There was one species, which is the major component, that when we compared the position with wild type ubiquitin here in red, then it was very similar in terms of peak location, except around the phosphorylation site, which I think makes sense. But then in the second form, which was more of a minor component, it had a big chemical shift perturbation in the C terminus. So obviously the phosphorylation had quite significant structural consequences. We wanted to study that a bit more. Crystallization of phosphoubiquitin gave the same overall fold as wild type, except for this patch of negative charge. Our secondary structure analysis showed that the fold was maintained in the major and the minor species. Our dynamics analysis suggested that essentially they've got the same rigidity, apart from at the very C terminus, which is more rigid for the minor species. We wanted to know maybe these 2 species were exchanging with each other, and we could analyze that by NMR with this ZZ exchange experiment, whereby we could see the exchange of one state to the other with a special experiment and then fit that and then tell you that they're exchanging between each other at a rate of about twice a second. Then we could use another NMR experiment. This is a long-range HNCO experiment to analyze the hydrogen bonding network in the system. And we were able to see that as we move from the crystal structure, the major form has the same hydrogen bonding pattern as wild type ubiquitin, but the minor species has a different hydrogen bonding pattern. This is shifted by 2 residues in this hydrogen bonding pattern, which means that the C terminus has been retracted into the core of the protein. And it's able to do that because of this leucine X, leucine X, leucine X, leucine motif, which enables the C terminus to slide in and out of the core of the protein. Not only does the C terminus retract, but it also exposes this phosphorylated loop to the solvent. And we can have a look at that with another NMR experiment, which is called CleanX. It's a solvent exchange experiment where we can see, for example, in the minor form that we have for this E64 in the major species is protected, but in the minor species with this extended loop, it's free to exchange with the solvent. So, phosphoubiquitin is a metamorphic protein, and you wouldn't have been able to see that in any other way except with our NMR experiments. But we wanted to know if phosphorylated ubiquitin is a metamorphic system, is ubiquitin a metamorphic system? We can have a look to see if we can see that too. So we can do that with an experiment called ZEST, which is chemical exchange saturation transfer. Very briefly, what you do is you have like a standard HSQC, but you add on to that a saturation pulse. The saturation pulse reduces the signal intensity. And I'm going to move the position of the saturation pulse from the bottom of my spectrum to the top. And then if I hit my peak of interest, then the signal intensity disappears. That's what you're seeing here. And then it should recover. If the ubiquitin is adopting a second state that you might not see, you can artificially prime the invisible state during the exchange process. And then you get a second dip in the location of the invisible state. And that's what's happened. That's what we saw in ubiquitin. So indeed, ubiquitin is a metamorphic protein, and we can fit this data and tell you that wild type ubiquitin is exchanging between this and the minor retracted species about 62 times a second, with a population of about 0.7%. The higher the temperature, the higher the population. And this retraction is important because if you look at the model this is a crystal structure of the kinase pink that phosphorylates it. the active site is really deep in the core of the protein. So if your ubiquitin can't retract, then it can't get that loop into the core of the protein, so it can't be phosphorylated. So it's functionally important. Just very quickly, the last 10 minutes or so, I want to talk about IDPs. So this is a project where we looked at the IDP tau with Sophia from the Scherer and Goddard group. Now, she was interested in how tau gets to its final paired helical filament structures, and she did this really beautiful study where she did a time-resolved cryo-EM and saw that at the beginning of filamentation, the structures always form this first intermediate amyloid called the FIA, and then over time they mature into the structures that you find in diseased brains. But they are really interested in understanding what is it about monomeric tau that might mean it causes the forms of FIA in the first place. So we're looking at the structural properties of tau. We can collect our HSQC, we can look at the secondary structure propensity, and we can see that there are regions in tau structure that have a preference for an extended backbone. So they're not completely disordered. There are some regions that have a preference for forming like an extended structure. We can then collect our standard dynamics experiments and see if there's anything obvious here. We can collect it at multiple fields, which we've done, and then look for any correlation. These are the residues that form the FIA. I mean, I stare at NMR data all the time. I think I can see there's definitely something going on in this T2 data, but it's not obvious, and I've got a lot of data, and it's not so easy to interpret. But we could try and dissect our multiple timescales of motion, and we can do this by looking at the spectral density. I don't want to say too much about this, but just to say, do you remember before I said that T1 and T2 sample motion on multiple timescales, unlike the heteronuclear NOE? Well, if you collect data at multiple fields, you can then extrapolate specific frequency-specific motion. And so the more fields you probe, then the bigger the window of frequencies of motion. But these data sets are still really complex. Can we distill it down a bit more? Well, we found this technique called impact, where you can basically work out what's the minimal way of describing the motions of a system with the spectral density data. So you can identify the bounds of a correlation time. Remember, IDPs have multiple correlation times, and you can say how many different correlation times you need to fit that model. And you do this analysis with the Akiyike information criterion that says, well, for this particular example data set, the IDP is best described with 6 different correlation times, and the bounds of those correlation times are between 20 nanoseconds and 20 picoseconds. And you can see for this structured part, you've got basically predominantly 1 correlation time, but in this disordered N-terminus, you've got contributions from multiple correlation times. And if we do that for tau, we can say that for this system, that data is best described by 5 correlation times within the bounds of 36 picoseconds to 36 nanoseconds. And what I think the main take-home message is, if we look at the slow motion of the tau molecule, we can see that the FIA residues particularly are experiencing a greater proportion of the slow motion. It's only a very small population, but there is a FIA-related structural dynamic difference. So these residues are experiencing more segmental motion. They're in a structure that's much more similar to the FIA that we see in the time-resolved time course. So you can imagine that energetically, it's much easier for these residues to start to form the FIA than perhaps a completely disordered system would. Very quickly, I talked about phosphorylation with ubiquitin, and phosphorylation is a really interesting biological process, but when you're studying it in the lab, it can be quite difficult to control. Tau in filaments is heavily post-translationally phosphorylated, especially in terms of its filament formation in a diseased state. But when you want to study that, that's quite difficult. to get a homogeneous sample. So in a follow-up study, Sophia created a PAD12 tau with phosphomimetic mutations. So that's you mutate your serine or threonine into aspartic acid or glutamic acid. That way you've got full control of your sample. Each residue that you want to be phosphorylated is affected. So what Sophia found in her analysis is that, so in the tau data I showed you before, this construct 297-391, it forms paired helical filaments. This longer construct, which is C-terminally extended, 297-441, it doesn't form paired helical filaments. So something about that system means it's inhibited. But when you have your PAD12 mutations, you get filaments again. So we wanted to see if there's anything we can tell by NMR. So we did the NMR assignment. You can see the difference between the wild type and the PAD12. There are peak differences. But what was really interesting was we look at the peak intensity, the peak height of the sample, and we compared them across the 3 different states. So in this 297-441 sample that doesn't form paired helical filaments, it's got a lower peak intensity than the 2 samples that do. One thing we noticed, that near the mutation sites, we have a second IVYK motif that also appears in the Thea sequence. So Sophia thought, let's mutate out that IVYK, let's get rid of that IVYK sequence and see what happens. We studied that by NMR. After Sophia pointed out that in the mutation, you can now get paired helical filaments, and these are the structures. And when we look at the NMR data, we can see that the blue one with the truncation has the same similar peak intensity to the samples that form filaments to the one that's inhibited. And what's really interesting is, you know, why would mutating something down here affect something up here? It must be because there's an interaction, right? And so that's led to this hypothesis that in the 297-441 system, there is an intramolecular interaction that inhibits downstream filament formation. But when you get phosphorylation, this interaction is disrupted, and then filament assembly can proceed. I promise this is my last slide. As a general outlook for NMR, we've noticed that, you know, applications of AI to our data analysis, one of the big things is looking at taking out the human element of the data analysis. And there is now work from Peter Günther, who has developed these Artina deep learning algorithms that are now available for everyone to use on this NMRtist cloud platform. And what they're supposed to do is you give them your raw NMR data, don't do anything to it, give it your sequence, and they'll pick all the peaks, they'll do the chemical shift assignment, and they'll even calculate the structure for you. So what we would envisage this to be a really good use for is validation of any AlphaFold model that you have with experimental restraints. Say you have a mutation that's structurally significant, functionally significant, that might give you a different structure. It would be really good to validate that structural change very quickly with this technique. It's supposed to take hours, not weeks and months as before. We're testing this in-house at the moment. We've got a few data sets, but what I'm saying now is we need you and your samples. So if there's a small domain, there's a small protein that's got a mutation that's functionally relevant, and you would like to see what difference that is, then we would really love you to come and have a chat with us, because we really want more data sets to check this. And then I just want to finish there. Here are the main references for the data that I've showed you. The NMR facility is me and Stefan and Trevor, and obviously we work in close collaboration with labs that use the NMR facility. So I'd just like to acknowledge those guys here, and thank you for listening.
Yeah, thank you, Steve, for inviting the EM facility to present here. Yes, we think we are one of the techniques which you can count as a biophysics. Yeah, I remember. but yeah, it's one of the microscopy technique could be used for see the molecule, because we say we're going to see, so this is really see, just not calculation. Also today is an introduction, so we welcome you to use our facility and discuss with us to see what we can help. And yeah, it is quite a hot area for this structural biology, and hope my introduction can give you some information. And yeah, this slide that we prepare with Giuseppe, which is my deputy head of the facility. So if you have his here, I decided I give a talk, and if you have questions, ask some sort of his project, there's an example there, then he's there to answer. Okay, let's go to the next one. Yeah, I want to give you some fact as advertised. So this is from the PDB, the Protein Data Bank. Yeah, every month they update almost every week. Yeah, this is I took yesterday. So you can see there's a different. This is the structure deposit into this database with 3 major technique or method to obtain structure. They also come as a combination. This is a multi-technique one, or Newton, or if you use Newton diffraction, they also count in. Here's a Newton, and this is a combination. So this is the timeline of year. So we are here now. So, but you can see EM is the yellow, and then the X-ray is the red one, and NMR the purple here. You just hear today all the talk. So we can see is from the beginning of the year 70s, the dominant method of structure determination is from there has been the X-ray crystallography. So they are doing very well and still continue to still keep on the top. But you can see our EM, they're very, very low. But from 90s, then we start to take off, and you can see they're up and down, but and also go to recent 10 years from here, like 2014, you can even get a higher speed. It looks like there's another no long, like a few years, you will take over to maybe take over the X-ray, because the X-ray seem to they just keep like that, did not go up. Here still EM still go up. In our LMB is different. I think from the Richard Henderson show is a few years ago, we only take over the X-ray. So this is just to indicate some factor of people use this technique more and more, and also 2/3 of the group are using we are using our facility. So I just want to amplify this part to also show the timeline of the LMB's EM in the next slide. Yeah, so I cut this from, for example, the year 1999 to now, and so this is cut. So I add some text to it, so we can see what our facility is expanding and the research in LMB to contribute to this takeoff of the electron microscope approach for structural study, for structural determination. So on the year 2000, in fact, 1999, the LMB facility, big change, renewal of the facility. So the order, like a 3 big order of the Tech9 microscope. This is a Philips, or and later called FEI. That time still called Philips. So from the number you can see 120 kV, 200 kV, 300 kV. So it's a big money, and also it is quite a new one. At that time, I still in Imperial College. We know that the volume here. So, and then you can see the EM structure really gradually come out. This is not just from LMB, from everywhere. It's gradually used more and more. And from, well, of course, I come from this point. I come to join LMB at this point. I don't know, somehow it's deep. But no, it deep. I can, yeah. And then we make it up again. Sorry, not me. Okay, so and yeah, well, this few days. So after a few years, and then we buy our first Polara 300 kV. This is before clouds, before the microscope we're using now, but this is our working horse on the 2007. And from this one, a few group in LMB start test the detector, direct electron detector, and source come in our same office writing his program. And this we got all the year here. And on here, 2013, we move into LMB. The new building here, very nice. And then we buy the 3 more microscope to here. This is our first class 300 automatic cryo-EM. At that time, for 4 million, not much. Now you can have the now the class 4 until here already 6 million of pound. So for when we have it. We have this renewal of facility also indicate this field going to the second stage with the detector, like direct electron detector become commercialized, and then we all the microscope same time improve the vacuum, reduce the contamination, and increase automatically data collection, this kind of thing, and plus this powerful image processing software is really become the speed of this data accumulation is really a lot of fold improved. So we all know we don't need to stay there one day overnight. You take one box 56 of film, and then you take 2 hours to develop film, and next day, and then scanning, and a few months until or like at least a half a year, you start to see what the structure is. Now, what we can do overnight, overnight data collection, no need to go to dark room, and then direct go to computer. And the first one is a new user, never learn microscope, come to use training. In 3 months, she got the data set, and in 6 months, published in Nature. Yeah, you know who. She is our star of like a ribosomes project, our favorite sample. So we know, so now it was really a golden stage of the cryo-EM. So of course, resolution is this is what the ribosome, like from our group Venki, and then also another few the membrane protein in the wall. So they just beat the 3 extra resolution. So this is what they call the revolution. So and from there, and then we the Nobel Prize is 2027 for recognize this achievement. So yeah, Dr. Henderson and with another 2 have this award. So it's it's very good for the EM field. And so from here, with some technique of the methods for who contributed the development of this field, like in here, the gold spot grid, this is the crystallisation and low risk group. In the 2014, they already experiment different support. So this is try to make the sample very stable. When you image, if you any of the blur, then this really affect your final resolution. So and until another few years later, they develop more like a graphing support one even better, and the graphing or then continue develop like hex gold. They become a gold standard of the most stable support in the research world, and also the expanding the 100 kV EM, and the detector become better and better. So in here, I did not write down yet because we haven't buy the new microscope. There will be a liquid helium, not just liquid nitrogen, liquid helium or phase play, or more even have a more better like electron lens, and like aberration correction, like CC, and then also CC corrector, CS corrector, this kind of microscope to speed up this field. Another one I want to point out is our FIP SEM. The FIP machine, since about 10 years ago, we have the SIOS, it's a manual one. But so it's slowly developed, not very quick, until 2022, Tame group come. So we have a first automatically a FIP machine, like from Zeiss crossfit. And then last year, past 2 years, we have Equilos, and this last year we have Actis. So it's a new generation plasma big machine, so it can do automatically like the cryo's, the 12 years ago introduced. So we hope this is another takeover of the not takeover, like another takeoff of this type of specimen. Yeah. So yeah, this is to give some fact of the cryo-EM. And the next one, yeah, this is the Nobel Prize. So I think you all know, yeah, 2017, yeah, Richard Henderson from our group here. Yeah, he also, in fact, he set up this EM facility at the beginning. So he's working on the direct electron detector. Yeah, use our CMOS detect technique to can record electron image. So in this way, we really the contact field have to shut down. Yeah, nobody use the contact field because everyone use the digital. The scientific application is the last people to use field. But and from 2013, our cryo-EM come. There's a few camera underneath, but we not use now because the detector already have a higher DQE than the film now. So this is what the revolution for. And also, well, there another guy, you know, the American professor who write the first image processing software. Yeah, not rely on. Yeah, it's a spider. And then this is this guy, Joe Show. Yeah, they all come here to give a talk. How to invent it, to freeze the sample without making the ice crystal. So this is what the 2017 is very nice. So I think you almost more or less know what is cryo-EM. So this is just a schedule. So looks very simple. You have a sample, but sample needs preparation in the tube, very pure, and then you put on the EM grid to make a one layer of liquid. And so here we omitted anything. It's like you need quick freeze to the liquid nitrogen temperature, and then you go to cryo-EM to take picture. So this cryo-EM picture very fuzzy, very low contrast. And now, of course, computer data processing, then 3D reconstruction, and then you build a model, and then you can fit your, then you can do a structural analysis. So this is what is cryo-EM. What is it? In a simple way. We were talking about this, the workflow in the next. But here, just an introduction. What kind of structural scale we're working on? Yeah. There are so many techniques now. This today you learn about. So the cryo-EM is these two parts, because these two main approach called the tomography and single particle. So you can see we are in this level. So from what the tomography is just like a light microscope, but becomes slightly higher resolution. So from micron, but you cannot go to very close the single, the like near very like near 1 angstrom. I think it's still difficult to see, but the single particle is approach to it. Is that we cannot single particle analysis cryo-EM cannot look at too big. This 100 nanometer almost a limit for electron 300 kV to penetrate, but it's also but the very small one, the single particle also cannot see. So the very small, like we say now, before we say 50 kilodalton. So now we can, some people like 30 kilodalton can achieve now. But the smallest we saw the people achieve is use what's called scaffold, like they build a protein fold and then attach the protein to it. In this way, they can look at the 14 kilodalton. This is from EVAC with the seeds. Is lyso side, yeah, the sample of crystallography, the most favorite sample. Yeah, they can attach it and then show to better than 3 angstrom resolution. Well, this is one kind of direction. So it's really we are approach to near 1 angstrom. But because this is the domain of X-ray AMR, you has been occupied this high valence domain. The cryo-EM is try to put the depth on that. Of course, for EM, electron microscope to resolution, ultimate resolution, of course, much better than 1 angstrom. Yeah, so 0.6, 0.7 when we do the Young's fringe. So we can, if we have a favorite sample, for example, a gold or a material, very thin, you can see the column of atom. So there's no problem for the microscope have this power, but the problem is at sample. The biological sample very short, very easy to get damage, and very low contrast because they are very light element. So this kind of all the limit, set the limit to the cryo-EM. But the modern technique, like the cryo-EM, now they frozen the sample. This is reduce radiation damage to improve resolution. So we think it still have much space to improve. So why we use? Yeah, now this is not just very like exhaustive thing to compare, but we just try to point out some advance of use here. So you all know. So for the cryo-EM, we can look at the membrane protein. This is very difficult to crystallize. We don't need to do the crystallization. So this time to make sample much simpler, shorter, and we don't need too much sample. Don't need very high concentration. So very tiny one. So for this type of sample is really ideal. So the cryo-EM really start from ribosome, single particle. And for 2D crystal, like bacterial adoption, because they have a naturally crystal, of course, you can use like electron diffraction. But this electron diffraction by the electron microscope, in fact, the type of crystallography. So we cannot really count. So we say we can look at the sample in this range. We don't need crystal to start with. So we also, because you got the water around frozen, so it's in a more like like a physiological condition. So no shrink or chemical fix to damage. Of course, we have a limitation that we write down here. Right, this is a bit out of date, 100 kDa. Now we are much push this. So it's not comparing with it, just you're talking about the one sample you are just here, crystallography or MR. They are very good at the small weight molecule. I talked to them yesterday. So what your advantage of X-ray? Yeah, so he just very good answer is now a lot of drug development, the pharmaceutical company, they just use the X-ray crystallography for screen the compound, but just because it's very small, and then you got a crystal already have structure. There's 8 different compounds to it to see what change it, which one is brought in. Yeah, this is the way to define the like called structural base drug discovery. So it's still very useful. So yeah, so we have different sample to shoot for cryo-EM. So now our facility, yeah, one of the many facilities in the LMB. So the people use for 24/7. So like because our book is on all the time, so anytime you come here, the microscope is collect data. So we have a 13 transmission electron microscope, include 4 Titan. This is a very high resolution data collection. We have totally 4 FIP. Yeah, they include the latest plasma FIP actis. We also access to the Hydra, another one, which is Routers group have. So total 17 microscope. With a lot of equipment for prepare sample. We have 170 users registered in our calendar, and so about two-thirds of those group use us. So now we have 7 staff. Yeah, that's a busy one. So this is what the technique we involve in. So like quite a few, huh? So now, yeah, what in detail, what we're doing is we provide training or training the user. Of course, LMB have a tradition of hang-on. Yeah, all the user, all the scientists have to learn what to do from very beginning to the finish. So it's a very good chance for you to learn this technique. So training Second is support. Yeah, of course, from the design of your experiment to how to set up microscope or troubleshooting during the data collection, and then later when you have problem, what goes next. So they're we all can provide. And we have a very wonderful EM wiki, have all the information for even to how to operate the microscope. Also in the also had a troubleshooting page. This is recent 2 years, very useful. So you don't need to really call us at midnight, say, oh, I what to do. You look at it first. Usually you'll see the answer. But of course, some of our people, it is answer as 24 hours. Okay, then maintenance. Yeah, make sure they're all working, update new microscope. We also do the research to like set up like a more the new development the novel approach, and also participate the research like we mentioned about like the other facility too. So if how are we going to start? So just go to our wiki, yeah, our wonderful wiki. You go to a new user. So this tell you step what we're going to do. So basically it's on this circle, yeah, from this clockwise. From here, you go to online, yeah, wiki, online, fill your form, of course, let your group leader know, yeah, she has the group. And then you will go to ask discussion, register our booking calendar, and then you also have to finish your safety training, liquid nitrogen, etc. And then you have to finish the training. Then you got the booking right to the microscope. We have 16. Yeah, you can choose. Also include the sample preparation. There are a lot in the calendar. Then you also can get help, or you can shadowing your lab people. They're already training. You just look at how they do your own sample, and then look at how they collect data. This is a very good way before you really go to a formal training. Or some people already can jump to start, so they can. So it's a shortcut. Then we are supported during your session, even you pass your training, and then afterwards we can also discuss for identify problem and change some kind of any feedback, this kind of thing. So yeah, we do this at our how you become used. So welcome to use our facility. So we got a very good program. This lab every 2 years we have the advanced EM course. They're all group leaders to come to give a talk and include us to do data collection. They're all in website, so you can start from look at the YouTube. This is to give all the information. It's very nice. So these are people. This one you can scan and immediately look at it or look at it from your mobile phone. And also there's a lot of training source. This is also in from our website. This is resource can be used like our course and some other way people's course. So very useful. Just get one, take one, and get started. So what do I want to explain? What we are going to be trained for? Yeah, this is a simple single particle cryo-EM workflow. Yeah, from sample to structure. So in this part, this is our most EM involved. Of course, you make your purified sample in your lab. This is your job. Then when you go to making EM grid, we help. We provide training. We help. Because this training, if sometimes you do not have time or you can know people to start, you can just give a sample. We can do for you. This is some kind of service or some kind of collaboration. But we can train you freezing sample, data collection on the microscope, and also then you got a data set. So this is the processing point. During the data collection, we have online processing. If some people want it, we have the program which already start the processing, tell you what the sample look like. Or maybe you got a 2D classification to see where your sample really was the collection. Also, but the rest of them, after we got data, can handle and copy from our facility server. You will transfer to our scientific computing facility. This you can do image processing now. So yeah, this is you pick up particle, we make a 3D map and do a model. This is a big chunk of data analysis. Okay, so for sample preparation, we are in the second floor. There's a 2 room for it. This is one of the room. So we got look like the old one. Okay, so we got the Vichy box. This one to freezing your grid. We have plasma for a cleaner for the surface treatment. We have glow discharge surface treatment. There are a lot of coating as well. Carbon coating is for you make your own carbon film. Yeah. So we also for the second floor we have high pressure freezing and cryo sectioning. You can do this type of this is second floor in 200. So this is another type of training. Of course, afterwards you may need to go back to use the Vichy box. And there's one data collection on cryos. So this is a class TM. So you will learn how to load sample, is how to load how to screen the grid, how to set up the program we call EPU to let you automatically collect data, and you to select your parameter for the dose, everything for you how to collect your data. This is depending on what you want to see. And so this is the energy filter and camera. We show you how to make use of this. So yeah, this is just a procedure. Your sample will look like that. This is your sample on the grid, and then this is a 2D projection from the beam. So your image will look like that. Black is the image, the gray is eyes. So in here, just a simple processing procedure. So we just see the particle on your grid is like that, and then the EM is a 2D projection from that's one. But this 2D projection, because the image formation, this 2D projection, in fact, is the 3D image centralized, from the Fourier domain, is the central slice of this object. So if you take a picture on this projection, this image, you end up that because the molecule is tumbling in the ice or before you're freezing, so you got different projection of the image. So in fact, this is when you can find your common frame and back projection. Use a mathematical way to back project to a common frame, and then this is come back to 3D. So this is the principle of how you from 2D image to 3D, because the 2D image already contain all the most of information. If you get enough projection, like enough copy. So this is the single particle, not involve any tilting. You just look at the image and do the picture. Of course, another approach called tomography. That means that's one we do not have enough. So there you have to enough projection. Then you have to tilt the stage. So but the end is the same. You go to build a 3D structure from the 2D image. Okay, so we magnify. Okay, this is my favorite one. So you don't need to sample, and then you got projection here, and this is what you got from the microscope. So this is a famous textbook picture. So why you got this? It's like you got a ship and you projection one way, you like a hand. Yeah, you cannot distinguish what it is. But if you this ship, if you go turn your camera or turn the ship, you got different projection. Now you will see the ship. Then eventually you will got the construction similar to this. Not quite same, because there are some missing data. You cannot exhaust the tilting. You always have some missing data or missing weight. So okay, so yeah, so I will show here. So this is also another schedule to like a diagram to show how you get back to 3D. Yeah, I think we need. Yeah. So when you get the different projection here, the computer program have to do alignment to find out the orientation related to the common frame. So then projection back. This is the model, but if you got a model of different projection, it's like you can see here. So this is the original image. So you are going to build this one into this one, and then calculate back, repeat to get a model more like the real image. So yeah, this is Giuseppe's project. Very good. This is some kind of enzyme. He take the picture and do the processing. First, you do 2D classification. Just put all the similar orientation to one group to improve the signal to noise ratio. Otherwise, this is a good image. Normally, it's very, very noisy. You cannot really see the particle, but this is one of the good, very good one. But it's very under focus. This is what we need to do different focus. So, and then this is the structure he get. It's very nice. And well, he fitted the backbone to he built the model, fit the backbone to the density. So this is to the cryo-EM map. It is a very wonderful one. This is a collaboration with source and then other people. So the next big one is the focused ion beam. As I told you, focused ion beam is one of technique to cut the big tissue or cell into thinner one. And the tomography is how you take the picture. You either turn the organometer to take picture, like you go to do your CT scan in hospital. Yeah, so you turn, or MRI, just turn the camera. Your people sit there, don't move. You're not going to turn patient, you turn the camera. Here, we can turn the sample. We not turn the microscope, not camera. We leave the microscope and camera, no. And then but we turn the sample. So but the similar, we get similar picture. So because the electron cannot penetrate to thick sample for 300 kV, this one cell it cannot penetrate. So what we do is you freezing it and you cut one piece and you look at it. Okay, but how to cut? We use this focused ion beam. Yeah, traditional you use room temperature, use a knife, diamond knife, or freezing use diamond knife. We still do it, though you can freezing use diamond knife, but there are a lot of disadvantage and difficulty as well. You can squash and then mark a lot of mark of the sample. But this is use ion beam in the microscope in a SEM, scanning EM, and then just slide the sum off. And then pick up the thinner one. We still have to go to the big microscope to take picture. It's called the cryo-ET, cryo-electron tomography. Yeah, so just like you do the CT in the hospital. So yeah, this is I don't want to go to it because it's time. I think too big. So this is what you're going to what it involves. So SEM had the electron image. You got a fixed focused ion beam. Generally, we got eye is I don't know what kind of eye we are using now. Is gallium, and now it's argon or use xenon. So they are depend on what kind of source you use. This high energy of the beam will cut the cell and then make a thinner one. So this is the block for holding the grid. This is special grid for doing the fit trimming. So it's the same thing. You have to grow your cell in this one, in the EM grid to hold, and then you have the freezing. Usually you need high pressure freezing, and then come here and then go to the fixed SEM machine. So this is liquid nitrogen temperature, and yeah, this is what I told you when it's doing this electron beam and ion beam. So you will see this is how you log for million. And so there's a technique called what's called sub-tomogram, sub-tomogram averaging. So when you're doing this section, it's just like your light microscope. You see the cell, but there's some protein like ribosome or some kind of like cryo-EM, this kind around. It's a very tiny dot. You want to look at the particle or improve the resolution of this image. You can use the single particle method to average it together. So if you identify the particle, find out what the orientation, you can average and to improve the signal and the signal to noise ratio a lot. And the techniques were quite complicated. Yeah, will be when you take a picture, usually you culture the cell and then you are freezing and then you do the flip milling, this same thing to cut. Then you go to the microscope to do a picture. You have to tilt different angle, take a picture. So this is the diagram you should to see or to show how you take picture. You when you tilt at different angle, yeah, you can see different, right? And then this is the conjunction. The conjunction you use to the segmentation to make the structure. But if you want to see more clear what inside, this one is a mitochondria. I'm not sure, I cannot really see it. And then you can use single particle method to average it, to cut individual out from here, here, and alignment and projection back. In this way, called the sub-tomogram. You use a tomogram and then do the average. So in this way, and then you do the lot of classification, 2D classification, 3D classification to build the model. In this way, you go to the single particle approach. So, but improve the regional log. This is why some people can go to 3 and so on already. That's a very very good achievement. Yep. So I will end up here. So this is a triple reconstruction from this milling and tomography. You can see this look like a tube or comet. Wow. And then you can see Golgi. This is one tomogram, and this is the model that they judge from there. And yeah, this is another type of the protein that my glass cannot really see. And so in here, in fact, they try to identify the protein here. This is very difficult work. Yeah. Okay, so I will end up here, big long. So this is thank you for all this our group people. Of course, we have we have 6 people, and Dom is kindly help us. So we got how we can save about 7, and then we got Greg look after our detector and some kind of and our data transfer. So he's very very good. And then we have a science advisor of Dr. Andrew Carter. Yeah. So yeah, thank you.